Once upon a time, there was a monk named Mendel. One day, he planted 4 pea plants. The pea plants grew to different heights. What's going on? Thought Mendel. He also noticed that there were different colored peas. Mendel decided to investigate. He took a yellow pea plant and carefully crossed it with another yellow pea plant. The new peas produced were all yellow. Then he took a green pea plant and crossed it with another green pea plant. This cross produced a mixture of green and yellow peas. Interesting. Mendel decided to cross the 2 colors together and found they made more green peas than yellow peas. What's going on? Why are there so many green peas? And why do yellow crosses stay yellow? Let's take a look inside a pea cell. That's where we'll find the nucleus. Inside of the nucleus are chromosomes. A chromosome is made up of very tightly wound DNA. DNA is made up of genes which code for everything the body does. For example, there's a gene that codes for height of a pea plant and another that codes for the color of its pods. Did you notice how a chromosome is shaped like an X? That is because it's actually 2 paired sister chromatids. They code for the same genes. If we looked at the pea color gene on one chromatid, we would find the same gene opposite it on the other chromatid. These are called alleles. Homozygous alleles are when they are the same version. For example, they both code for a green pea, or they both code for a yellow pea. Heterozygous alleles are when the pea color gene is expressed differently on the 2 chromatids. Thought Mendel, green pea or yellow pea? Oh! Mendel realized that different alleles were either dominant or recessive. Homozygous alleles will always express the color they are. But what happens when they're heterozygous? Turns out the green allele is dominant, while the yellow allele is recessive. That's why the heterozygous pea will be green. Mendel pondered his pea plants. Was there a way to predict the next generation? Later, another scientist discovered a handy tool to answer just such a question. It's called a Punnett square. First, we label our peas with their alleles. We'll use letters. Because green is dominant, green peas could be hetero or homozygous. Let's cross 2 yellow peas first. All of the offspring are yellow. What if we swapped one of the yellow peas for a green pea plant? For this to work, we need to know the alleles. Let's assume we have crossed a pure homozygous strain. All are heterozygous. Let's take one and cross it with the yellow. Half the offspring are yellow now. And if we cross the 2 heterozygous pea plants, there's a 1 in 4 chance the offspring will be yellow. Neat! And that's Mendelian genetics.